Oh, hello there. Today I'm going to teach you about transcription and translation, which is the process by which DNA, or the genetic code of your body, is changed into proteins, which is the structure of your body. This is a model of DNA. DNA is made up of two sugar phosphate backbones, the sugar being deoxyribose, and in between are nucleotides, adenine, thymine, cytosine, and guanine. The sequence of these nucleotides in between the structures is what determines your genetic code and how you will look and do pretty much everything. During transcription, we want to protect DNA as much as possible because it is very important. To do this, we make a copy called mRNA, which we will use in the translation process. mRNA is very similar to DNA, except for it has a uracil instead of thymine, and it has a single sugar phosphate backbone, with that sugar being ribose instead of deoxyribose. Transcription begins with a very important enzyme called RNA polymerase. RNA polymerase separates your DNA and begins reading one nucleotide at a time. These nucleotides are built up so that a new strand is built of mRNA, where every time RNA polymerase reads an A, it will put a U, and when it reads a C, it will put a G, T, it will put an A, and G will put a C. This is very important, and this is where A, T, C, and G stand for the different nucleotides that are possible. As this is built up, where it starts is at the top box, which is a string of T's and A's, and it will end once it reads the sequence AAU, AAA. Once it reads that, it will have built up a new strand of mRNA that is the exact copy of the important part of your DNA, which will then be translated into a protein. And here we can see that the sense strand of the DNA is copied by RNA polymerase copying the antisense strand, which is the non-coding DNA. So whatever is here is going to be built in to proteins, which is what your body will end up looking like, and everything that's important to how your body functions. Once this mRNA is completed, translation can begin. mRNA exits the nuclear pore from the nucleus and enters into the cell matrix, where a small ribosomal subunit will connect to the mRNA strand and begin reading 3 nucleotides at a time. With these 3 nucleotides are called a codon. When each codon will correspond to an amino acid, and amino acids, when put together into a polypeptide chain, turn into proteins. The very important ones are MET, which is the starting protein. When the small ribosome reads A, U, A, it will start coding and it will start making a protein. The way it does this is by matching up these 3-letter codons with an anticodon on a very important piece of tRNA. tRNA is a piece of is a structure which connects an anticodon to a amino acid. The amino acids will then turn into a polypeptide bond. So the small ribosome will find the methionine, which is the starting codon, and a tRNA will bring over and start building an amino acid. Once it has read the first codon, it will move on to the second codon, which here is asparagine, and it will find the corresponding tRNA, and it will continue to build up an amino acid as it moves along until it finds a stop codon, at which point the ribosome will break the amino acid chain and let the protein leave completed. Up next we have an animation of this process. Here we have the starting code with its anticodon. The ribosome, large ribosome connects and a polypeptide bond is built. This will continue along, building a more complicated chain of amino acids until a stop codon is found, which is the very last one here. Once that is found, a releasing factor will come in and it will free this polypeptide chain and turn it into a complete protein. This is the process of translation and finishes up the whole process of transcription to translation. Thank you for watching.